This protocol combines the bottom-up-based DNA origami technique with top-down nanofabrication methods, allowing the parallel fabrication of inorganic nanostructures with sub-10 nanometer feature sizes on different substrates. This technique can be used to create billions of accurate nanostructures at one time virtually in any shape without the need for expensive patterning methods.
This method has the potential to be harnessed in sensing applications such as surface-enhanced Raman spectroscopy, as well as to create novel optical metasurfaces. Although this protocol is rather straightforward, multidisciplinary fabrication skills are needed, and therefore visual demonstration will be useful for researchers with a variety of backgrounds.
Demonstrating the procedure with Petteri Piskunen will be Sofia Julin, another grad student from our lab.
To make the stock of staple strands, mix equal amounts of all of the oligonucleotides needed for the bow tie structure, and mix 20 microliters of the M13-MP18 scaffold strand with 40 microliters of the staple stock solution and 40 microliters of 2.5x folding buffer in a 200 microliter PCR tube. Then anneal the reaction mixture in a thermocycler from 90 to 27 degrees Celsius as outlined in the table. To prepare the substrate, immerse approximately 7 by 7 mm chips cut from a sapphire wafer in a glass container at 52 degrees Celsius acetone for at least 15 minutes before transferring the chips into a glass container of isopropanol for 2 minutes of sonication. At the end of sonication, use tweezers to remove the chips from the isopropanol.